Hello, students. Um, Hope you all are doing well. And today we are going to study the last session on this module. And today it is related to the topic of the diabetes. So today we will cover about prevention, preventive measures, and what is actually the diabetes mellitus is, and how it can be managed through diet and physical activity. So let's start today's session that is related to the module Nutritional Biochemistry for Exercise and Health. Today's session aim is to cover and discuss diabetes, its incidence and mechanism, and what are the diagnostic tests to measure and evaluate the diabetes mellitus, and what are the possible interventions to reduce the incidence of diabetes, along with discussing these interventions as well, along with and comparing with the lifestyle and interventions. So let's study about the diabetes incidence. Here is the link to study more in detail about it. It is estimated that in UK, that around 3.7 million of people have been diagnosed with diabetes, and when this all data was published in the year 2017. Right, and around the 12.3 million people are at increased risk of type 2 diabetes mellitus, whereas around 4.6 million people are estimated to live with diabetes, either they are diagnosed or undiagnosed as well. Among the UK, the prevalence of diabetes mellitus type 2 is higher in England, which is more than 3 million. Yeah. Now let's go to the facts and figures. What are actually the risk factors of the diabetes are? People, what we say that are more at risk of the diabetes type 2 rather than the type 1. Type 1 is actually the hereditary genetic type, while the type 2 is associated with our lifestyle and other factors rather than the genetic factor. And it is estimated that if our lifestyle, our habits will not change, so it is estimated that it will be worse, and around more than 5 million people will live with diabetes type 2 in UK by 2025. So here is the research and all things were discussed, and some links are here shared with you. Please have a look on this and make a note that what the risk factors you will find out from these pages. Okay, that are related to type 2 diabetes. So let's discuss it and then compare with this slide, your results, that what you find out. Yeah. So now let's go and first discuss what actually the type 2 diabetes is. Type 2 diabetes is related that high level of glucose in the blood of a person or an individual is called as the type 2 diabetes. But what this type 2 is associated related to the other factors, not the hereditary one that results to the hyperglycemia of a person. Hyperglycemia means that increased level of glucose in the blood of a person. And why this happens? Because in our body, pancreas, one of the main organs, is present that have some receptors called as beta receptors that produce a hormone called as the insulin. At the time, sometimes insulin do not release or do not work. The pancreas receptors are being affected. They are not working because of obesity or any, diseases or problem. Or the other thing is that because of the lifestyle. So these are the some factors that results to the type 2 diabetes. And how it's work. For example, we are taking a food in our meal. The most of the content on the carbohydrates that also converts into glucose or in the form of a sugar. So when glucose come into our body and digested from our stomach intestine into the bloodstream, when this sugar go into the bloodstream, it is needed to go inside the cells to function properly, whatever their function is. And how it can be performed by means of the hormone called as the insulin. So whenever the glucose enter in our bloodstream, this state is called as a hyperglycemia. Then our insulin is released by the pancreatic beta Langerhans cells, and why they are releasing? To help our body to move this glucose from the blood to the cells to perform their functions in a appropriate way. However, in diabetes, this function will not be performed, and in this way, this glucose level remains in our bloodstream and becomes more higher and higher. And this state is called as diabetes, because this is not a normal situation. And what happened in this result of diabetes, hyperglycemia, that cells will not function very well, and the other more chances towards infection, because most of the bacteria, other microscopes, they also survive on the glucose level, so they get a plenty of the glucose for to get the infection to that individual then. So this is the party for them. So it's like that. So let's have a video, watch this video, and understand this topic more. Now let's go to a research. Please, after this lecture, go and read this research as well. It's showing some basic concepts about the type 2 diabetes, that how it develops and how insulin resistance develops, especially in obese people, because of the layer that's made by adipose tissue, the extra amount of the fat, the nutrients that we are not burning, and it becomes into the fat and is stored in our body, especially around our viscera, that's mean around our organ and beneath the skin. So here are some key concepts that are being used in this research, in this article, that number one is the hyperinsulinemia. Hyperinsulinemia means that excess level of hormone insulin mistaken for the diabetes that is releasing in the body. Then the other term that is commonly used is the hyperglycemia. Hyperglycemia means abnormal high levels of glucose in the body of a person. The third another term is endogenous. Endogenous means that inside the body, it is released or present inside our body or generated inside the body. It's called as the endogenous. And where the hepatic term means liver, splanchnic means that the internal organs, especially that are present in our abdomen, like the pancreas, liver, that means the gallbladder. These are called as the splanchnic organs. And what actually they are clearing the concept of the diabetes too is that it is caused by a reduced capability of pancreatic beta cells that are present in our pancreas, and it have different cells that releases different type of hormones. In this pancreatic cell, the specialized cells called as beta cells, they produce the insulin hormone in response to the glucose intake when we take from our meal, and when it is digested and come into the blood, you know. Then in result, our pancreatic beta cells produce insulin hormone. And why this the release to regulate the glucose uptake along with the glucagon, another hormone, to maintain the glucose level in our body. And it is depending on the internal and external availability. Yeah. Now, next is about the influences factors. What actually are the causing factors that influence to cause the type 2 diabetes? So here are some questions that which are some physiological factors, our lifestyles, our dietary habits, and some related to psychological factors. So please make a list and discuss in the comment section and discuss with it, and find out what are actually the dietary recommendations that would you make to help to reduce the chances of type 2 diabetes developing. Here are some clues I'm giving, such as high fiber diet. There is another research that we discussed in the previous session that was related to the whole meal, such as the whole apple, or its juice. Which one is better than to maintain? So please put all these notes and let's discuss. Okay. Now let's go to the measurement ways that how the blood glucose, or we can say the diabetes, can be measured, evaluated. There are some laboratory tests that can be performed. Along with the number one is oral glucose tolerance test. What is mean by the oral glucose tolerance test is, first, take out the points from this main heading, this main point, that what is mean by the oral means that taking the glucose by mouth orally. So it's mean that oral glucose. And what is mean by then the tolerance test, that after taking this glucose by mouth, the person is able to tolerate in the blood, yes or no. So in the same simple terms, it means that what we are going to do, how we can assess the person's the glucose level in the blood. First, we will take a blood sample from that person, a blood test, and evaluate what the glucose level of that person is. Okay. After that, taking a blood sample and give them a bottle of 75 grams of glucose to take it orally, yeah, by mouth. When the person takes that glucose after the few hours that are required, then again take another blood sample, a blood test from the person, and then evaluate and put out the difference of the glucose level in the blood of that person before and after the exposure, after taking the 75 grams of the glucose, which will show the levels of glucose that either the person have the high glucose or the glucose intolerance or the type 2 diabetes or gestational one or reactive hypoglycemia is. So here these some special terms are coming that you may not know, that are gestational diabetes and reactive hypoglycemia. Gestational diabetes actually is associated related to the pregnancy associated diabetes. Those women who are pregnant, and in such women glucose levels are found higher, and it may be because of the changing in the hormonal levels. That's why it is happening during the pregnancy. While other is the reactive hypoglycemia, it means that a certain increase of the insulin. That's result. A person take the food rather than releasing of the normal amount of the insulin, pancreas releasing a lot of insulin, and it's result to the certain hypoglycemia of a person. Maybe person after eating may feel fainting or some fatigue, or maybe shaking of the head because of this reactive hypoglycemia, which is not a normal, physiology. Another test is the blood glucose levels by measuring through a machine that is called as the RBS, that is the randomized blood sugar levels. But the main appropriate is that is performed in the labs as well and by the machine as well, but after the fasting, around 8 to 12 hours. And what it result, then it shows the actual level of the glucose after the fasting, that is still higher level of glucose is present after the fasting, that it is also showing in the form of the zones in picture as well. So it means that the person is then suffering from the hyperglycemia, that is the diabetes. If not normal range of diabetes coming, then it means that the person is not diabetic, because after taking the food and the fasting zone, normal zone will come. And after this normal zone, of course, then the all glucose level will come into the cells. Then the state of hyperglycemia will not be occur, will not be finded out. If it happens, it means that the person is hyperglycemic. So go for these zones and find out the readings and put it in your workbook. In these zones, what you find out how much amount of the glucose millimoles per liter. Yeah, these are 7, 6, and 3.5, but find by yourself. These are the normal ranges that I shared with you. So go to the research article that I'm sharing with you. It is published in the New England Journal of Medicine with the title topic of the research is reduction in this incidence of type 2 diabetes with lifestyle intervention of metformin. Either the lifestyle intervention was successful or metformin. So have a read and find out what the task I'm giving to you. And task one, note it down that what the introduction of this research paper says. So make the points what actually the introduction is, right? Then in the task one, second point is that which racial groups in the paper are at particular risk, such as obese. Yeah, other are maybe psychologically related, maybe due to stress or anything. So please find out and note it down. Then the third point, that what are the primary research questions in this research article are. Now task 2, read the abstract of this article and take out the main important points and what were the main findings of this study that were put in this abstract. Then last task, that is the task 3. Go for the figures in the result section of this article from figures 1 to 4 and what the key findings you have been observed in these figures and which of the interventions is consistently more effective in these figures. In the result section you find it out and please share with it which one is the best. One of these figures I'm sharing with you, which is the figure number 3. What is showing this figure, that the researchers observed 3 interventions to different groups, that these groups of the people were obviously diabetic. So who found more effective? First group was given the placebo. Placebo means that we give any tablet and say in a form of a tablet, but it was not treating anything, nothing. So that's why we are saying it as a placebo. So what happened after giving placebo to that group? See in the graph, nothing was observed. The glucose levels were still getting higher and higher. Okay, still by taking the placebo. So no effect of placebo was on this group. Where the other group was given metformin, a tablet that decreases the glucose level to treat the diabetes to such persons. Here in this figure, we find it out, we realize that yes, metformin, this drug works to decrease the glucose level of such group of person. However, the third group, which was given the intervention, that is by modifying the lifestyle, were more successful to decrease their blood glucose levels and diabetes risk by adopting this intervention. So now you can realize which intervention is more successful, whether the drug only or nothing only, that is the placebo, or modified lifestyle with physical activity and diet. And the third last one, according to another research, further research was going on. And what they realized, that if the person is still a very dependent and have severe diabetic, so these modified life changes along with the metformin or any hypoglycemic drug gives very drastic and significant good results to control the diabetes and hyperglycemia in such group of people. So please find out the evidence, put in here, and compare with the studies that which is the most successful intervention to control the diabetes and hyperglycemia in such population groups. Now let's go to the summary of today's sections that we discuss what is the diabetes, what is actually the mechanism of the hyperglycemia, how much the prevalence and incidence rate of diabetic cases in UK are, and what are the diagnostic procedures to evaluate diabetes in laboratory or by means of the home machines. Then lastly, we discussed about some possible interventions to reduce incidence of diabetes along with discussing and comparing the lifestyle along with the drug interventions to reduce the symptoms of diabetes. Hope you will understand today's topic. If still you are not understanding or any questions, you are most welcome to ask. So anytime you can email me or ask me anything. And goodbye, as today is the last session of this module. Best of luck. Hope you will do all your assignments, all your tasks promptly and at time. Best of luck. Thank you.